Hello everyone, my name is Étienne. I'm a PhD student at University of Sherbrooke, and today I will talk to you about characterizing the regulation of the human snoRNome. So what are snoRNAs? snoRNAs are small nucleolar RNAs with non-coding RNAs that can be separated in two groups according to their structure and function. So we have CD-box snoRNAs and HAC-box snoRNAs. They both have a similar function, which is to guide modifications on their target RNA. So the canonical target of snoRNAs are ribosomal RNA. The modifications they guide on ribosomal RNA are highly important in ribosome biogenesis. However, several snoRNAs don't have a known canonical target, and they are thus called orphan snoRNAs. But in the recent years, several of these snoRNAs were associated with new non-canonical targets, such as pre-mRNAs and tRNAs. And with these new targets often came new functions associated to snoRNAs, such as the regulation of splicing, of pre-mRNA stability, and also of polyadenylation. Most snoRNAs are encoded within introns of a gene called the host gene. This host gene can be coding for protein or non-coding. So the expression of snoRNAs is depending on the transcription and splicing of the host gene, which generate a host gene transcript and a mature snoRNP, which is highly stable because of proteins bound to it and which can act in the many different ways that I told you in the last slide. So one big question is which factors modulate the expression of snoRNAs? So it's clear that at some point the host gene will be implicated, but the degree of implication of the host gene is not so clear. First, because a lot of snoRNA host genes were found to be highly susceptible to nonsense-mediated decay, and also because recently several snoRNA host genes were found to harbor dual initiation promoters. So these dual initiation promoters are basically a canonical transcription initiation site called the YR promoter, which is present in most human genes, and it also contains another transcription initiation site that is called the YC promoter, and this is thought to uncouple the expression of snoRNA and host gene transcript. So we can say that snoRNA and host gene relationships are not as simple as they look, or they are more complex. I found in the literature 3 studies where they compared the expression of snoRNAs and their host gene at the same time. One was in blood samples from acute myeloid leukemia patients, one in human cell lines, and another in cell types from mouse. In the 3 studies, they found that there was a non-correlation between the abundance of snoRNAs and their host gene. So this raises multiple questions. One could be that how does the cell produce different levels of snoRNA and host gene transcripts from the same gene? Another could be that this non-correlation that we see between snoRNA and host gene abundance, do we also see it in healthy human tissues? So this brings me back to the title of my presentation, which is Characterizing the Regulation of the Human snoRNome, so basically all snoRNA expression. So I will have 2 objectives for my project. The first one is to determine the snoRNA abundance patterns across normal human tissues, and the second one is to identify the main modulators of these abundance patterns. The method we used is called TGIRT-seq, so it's an RNA-seq approach that uses a thermostable reverse transcriptase called TGIRT, which was shown to accurately reverse transcribe highly structured RNAs such as snoRNAs, but also other types of RNAs such as messenger RNAs. So we used the TGIRT-seq on 7 healthy human tissues using 3 biological replicates each. We first ribodepleted and fragmented the samples. Then we proceeded to the reverse transcription using TGIRT, and we sequenced the libraries, and we proceeded to the trimming of the reads, then aligned the reads to the human genome using STAR, and finally assigned the reads to genomic features using a tool developed by our lab called COCO. So for the first objective of my project, I used a metric called the coefficient of variation. This corresponds to the standard deviation of the expression of a snoRNA across tissue divided by the average abundance of that snoRNA across tissues times 100. So to give you an idea, a low coefficient of variation corresponds to a more uniform expression across tissues, whereas a high coefficient of variation corresponds to a more enriched expression in either one or a few tissues. I represented these coefficient of variations for all expressed snoRNAs in the form of a density plot. So each line represents here one snoRNA, and you can see that the snoRNAs tend to separate in two groups or classes. So I calculated the derivative of this function here. I traced the tangent at the point where the derivative was the most negative, so this gave me a threshold here. Below that threshold, we had then 390 uniformly expressed snoRNAs, and above that threshold we had 85 tissue-enriched snoRNAs, so two abundance classes for our snoRNAs. These tissue-enriched snoRNAs were mainly enriched in brain and also in reproductive tissues. I then looked at the correlation of abundance between snoRNAs and their host gene. So we can see that most snoRNAs are positively correlated with the expression of their host gene, which is logical if you remember my introduction, where I told you that the expression of snoRNAs depended on the transcription and splicing of the host gene. But interestingly, about 40% of snoRNAs are either non-correlated or anti-correlated with their host gene abundance. Looking now at our two abundant snoRNA abundance classes across these correlation of abundance, we can see that tissue-enriched snoRNAs are mostly positively correlated with the expression of their host gene, whereas uniformly expressed snoRNAs tend to be more non-correlated or even anti-correlated with their host gene. So this ends the first part of my talk. So what I want you to remember here is that in normal human tissues, we have two snoRNA abundance classes. The first one is the majority, uniformly expressed snoRNAs, which are mostly anti or non-correlated with their host gene abundance. And the second one is tissue-enriched snoRNAs, which are mostly positively correlated with their host gene. Now for the second part of my talk, so I first looked at the host gene biotype, so whether the host gene codes for a protein in purple or is non-coding in pink, dividing by our two abundance classes of snoRNA again. You can see that in both cases, snoRNAs encoded within non-coding host gene tend to be positively correlated with their host gene, whereas when they're encoded within protein coding host gene, they tend to be more non-correlated or even anti-correlated with their host gene. Next, I looked at the susceptibility of host gene to nonsense-mediated decay. So we can first see that anti-correlated non-coding host gene of uniformly expressed snoRNAs tend to be highly subject to NMD, whereas the more the non-coding host gene is correlated with its embedded snoRNA, the less it is subject to NMD. So this suggests that these anti-correlated non-coding host gene here serve the only purpose of expressing the snoRNA they encode, whereas the rest of the transcript is then quickly degraded by NMD. Whereas here, these host genes serve yes to produce the snoRNA they encode, but potentially serve another purpose in the cell as long non-coding RNAs. Now for a non-coding host gene of tissue-enriched snoRNAs, none of them is subject to NMD. And for protein coding host gene, we don't see a clear trend across correlation of abundance. Finally, I looked at the presence of dual initiation promoter within a host gene to see if it could affect the correlation of abundance. So we can see that for a dual initiation promoter host gene in green, they tend to be more non-correlated or anti-correlated with their embedded snoRNA compared to a simple initiation promoter in magenta. We can also see that dual initiation promoter host gene are highly subject to NMD, which is less the case for simple initiation promoter host gene. And we see similar trends for host gene of tissue-enriched snoRNAs. I was then really interested in that group of snoRNA which seemed to stand out of the rest. So I looked at these snoRNAs and they are all highly enriched in breast or ovary tissues. So to conclude, I would like to present you a model in which it all starts with the snoRNA within its host gene. The first big separation is whether the host gene codes for protein or not. We saw that most protein-coding host genes are anti or non-correlated with their embedded snoRNA, and that they mostly harbor dual initiation promoter, which gives the opportunity to create an alternative initiation of transcription. On the first case, it would generate a functional host gene transcript that would be translated into a protein and a snoRNA. In the second case, it would generate snoRNA, but a host gene transcript that's degraded by NMD. In both cases, it would produce a uniformly expressed snoRNA or a breast/ovary enriched snoRNA. For non-coding host gene, we saw that most of them were positively correlated with their embedded snoRNA, and that through simple initiation of transcription, it would always generate a functional long non-coding RNA and a snoRNA that's mostly a tissue-enriched snoRNA. But the take-home message here that I want you to remember is that the use of dual initiation promoter and NMD allows the cell to uncouple the expression of snoRNA and host gene, and this ensures a regulation of expression that is specific to each different tissue to generate specific level of snoRNA and host gene transcript in these tissues. With that, I'd like to thank my lab and the funding sources and you for your attention.